Okay, so as they're returning to their seats, we'll move on to the next section so we don't get too far behind, which is pharmacovigilance in the electronic health system. Our first speaker is Jessica Weintraub. She's in the Division of Pharmacovigilance at the FDA.
Thank you very much. Today, I'd like to describe tools and processes we use at the FDA for detection of safety signals for SJS-TEN, and to provide you with a high-level overview of the regulatory aspects of adverse event reporting with emphasis on SJS-TEN. So FAERS, or the FDA Adverse Event Reporting System, is the primary tool we use for detecting safety signals for SJS-TEN in the post-marketing setting. It is a database of spontaneous adverse event reports that has been in existence under various names since 1969 and supports our post-marketing surveillance program for drugs and therapeutic biologics. In the reference slide at the end of the presentation, I've included a link for information on how the data files, which are publicly available and updated quarterly, can be downloaded, and how to request individual case safety reports under the Freedom of Information Act. FAERS data cannot be used to calculate incidence rates of any adverse event for a number of reasons. We don't receive reports for all adverse events that occur with a product. We don't know the size of the population exposed. And there are many factors that can affect voluntary reporting. The analogous database for vaccine adverse event reports is VAERS, and for dietary supplements, as well as foods and cosmetics, is CARES. There are only 4 required elements for a post-marketing adverse event report to be considered valid. There needs to be an identifiable patient, an event or problem, a suspect product, and a reporter. But a good post-marketing case report for SJS-TEN will include much more information. It'll include a description of the adverse event, the patient's background characteristics, allergy history, medical history, information on how the diagnosis was confirmed, biopsy results, a list of the patient's medications, including those recently discontinued, over-the-counter meds, herbal supplements, and so on, and the time to onset for each of these in terms of the onset of the event. The action taken for each of the products, for example, if all were discontinued at the time of diagnosis or if any were continued, and how that affected the clinical course. We also appreciate reporter contact information in case we need to contact for additional follow-up information. Sometimes additional information becomes available after an adverse event report has been submitted, which changes the suspect products or the diagnosis. And submitting an updated report can prevent incorrect attribution to a drug. There is now a mobile app called MedWatcher that enables you to report cases of SJS-TEN and other adverse events using a smartphone or tablet. And it's a free app that can be downloaded through iTunes or Google Play. This slide outlines the regulatory requirements for drug manufacturers to submit adverse event reports that they receive to the FDA. The first category of reports, the 15-day reports, are for those events that are both serious and unexpected. I'll get to the regulatory definition of serious on the next slide. But unexpected or expectedness is based on what is currently in the US label for that product. Manufacturers are also required to submit foreign reports that meet the criteria for 15-day reports to the FDA if they market the product overseas, and medical literature cases that they're aware of. Serious and expected reports, as well as the non-serious reports, fall under the category of periodic reports. And these must be reported quarterly for the first 3 years after approval of the product, and then annually after that. Per regulatory definition, a serious adverse event is one that results in death, is life-threatening, results in or prolongs hospitalization, results in disability, congenital anomalies, and the category of other serious, which, for example, includes things that would require intervention in order to prevent a serious outcome. And cases of SJS-TEN could fit multiple categories. Incoming reports to fairs, whether submitted directly through the MedWatch program or coming in from drug manufacturers, are evaluated by healthcare professionals, usually pharmacists, called safety evaluators. Each safety evaluator is responsible for a group of drug products, and new reports and updates to those reports for their products are pushed directly to the safety evaluators. We maintain a list of what we consider to be the most current, the most severe adverse events, and SJS and TEN is on that list to facilitate identification of those reports in the large volume of incoming reports. Because the information provided in spontaneous reports can be variable, we need to tailor case definitions to our data. The same goes for causality assessment. We use causality assessment, but because we often lack the information needed to calculate an Alden score, we typically use the causality criteria from the WHO Uppsala Monitoring Center. We also perform periodic product reviews in accordance with current regulatory guidelines. And one example of these are the pediatric reviews triggered by pediatric labeling under the Best Pharmaceuticals for Children's Act. When a safety signal is identified, there are a number of possible regulatory actions. Those in pink represent options for labeling under different sections of the label. SJS and TEN typically are labeled in warnings and precautions, although there are cases where it may appear in a boxed warning, and there are also situations where it may appear in the post-marketing experience section of adverse reactions. When we identify a safety signal, we can require the drug manufacturers to issue Dear Healthcare Professional letters warning the medical community of the risk. We can issue drug safety communications. We can use post-marketing requirements or post-marketing commitments, PMRs and PMCs, to further evaluate the event. In some situations, the use of REMS or risk evaluation and mitigation strategies may be considered necessary in order to manage a risk while enabling continued access to the product. And the option of last resort is market withdrawal. We also have access to VigiBase, which is a global database of spontaneous adverse event reports maintained by the Uppsala Monitoring Center. And VigiBase can be useful in a number of situations. For example, when a drug has a more extensive marketing history outside the US before approval in the US. The medical literature is another important data stream. As I mentioned, drug manufacturers are required to submit literature reports to the FDA that meet the criteria for 15-day reports that they are aware of. But safety evaluators schedule ongoing literature searches for their products to catch additional cases. This publication by Helena Pontes and others highlights some situations in which safety signals were identified, in some cases in fairly sizable case series, but were not reported to regulatory authorities. Another advantage of published cases is that they tend to be well documented, and these can be very helpful. The volume of incoming data is large. I think in 2016 there were about close to 1.8 million reports coming into fairs. So data mining can help us identify patterns of association through disproportionate reporting. We can use it in fairs and VigiBase. In the references slide at the end, I've included a publication that describes data mining activities at the FDA. We use it for hypothesis generation. It does not replace individual review of the cases because there could be duplicate cases reported by various manufacturers of a product, or there could be a drug that is frequently co-prescribed and may be an innocent bystander. NICE-CAIDS is a collaboration between the Consumer Product Safety Commission, the Centers for Disease Control, and the FDA. And it uses trained abstractors in a nationally representative sample of emergency departments to collect data on adverse reactions that are diagnosed and treated in emergency departments. Now, there are some limitations to NICE-CAIDS from our perspective at FDA. We get the data files updated for NICE-CAIDS on an annual basis, so there's considerable lag time as compared to FAERS. Also, the case reports from NICE-CAIDS do not include information that was obtained in the inpatient hospitalization. It only includes information from the emergency department. So biopsy results, even dermatology consults that were not available at the time of the emergency department visit may not be available. Post-market pharmacoepidemiologic studies involving prospective data collection, such as registries, can be advantageous in providing specific information such as ethnicity, genetic information, but there may be under-ascertainment of cases, and for a rare event such as SJS-TEN would need large numbers of patients enrolled. Studies using large administrative databases collected retrospectively provide a look at the use of the drug in the real-world setting with large numbers of patients, but there may be under-ascertainment of cases through billing codes, and I'm looking forward to hearing more about that in the next lecture. And there may be incomplete capture of some information, such as ethnicity. And finally, looking toward the future, Sentinel is an active surveillance system sponsored by FDA, comprised of primarily of administrative and claims data, with the eventual goal of incorporating more information, more clinical data, electronic health records, Medicare, disease registry data. ARIA, or the Active Risk Identification and Analysis Program, is a subcomponent of Sentinel that uses predefined modular tools that enables rapid querying in the common data model. So in summary, rare events such as SJS and TEN with relatively short latency periods, we use FAERS primarily for signal detection and identification. Additional tools are Vigibase, medical literature, data mining, and NICE-CAIDS, and US-based pharmacoepidemiologic studies and Sentinel are promising for the future, and research is ongoing in this area. Thank you.
Thank you, Jessica. I see that Josh has arrived. Your slides are loaded and stuff?
I think they are.
Okay. All right, good. We'll have time for questions at the end, and we'll see if Josh's slides show up. Josh is at Vanderbilt University in the Department of Biomedical Informatics and Medicine, and he's very involved in the Precision Medicine Initiative as well.
Great. Thank you, Jeff. I'm going to talk about some experiences that are built collectively at Vanderbilt around using electronic health records for research, and most of this has been done on common disease, and we're going to talk about some of the experiences on specifically around SJS and TEN and rare disease, building to that story. So first, as we have used electronic health records a lot as a platform for research, and one of the reasons for this is really a driving desire to leverage all the information we collect as a byproduct of traditional healthcare. And what we see here is just a little survey of Vanderbilt's the data in our biobank. So we have 230,000 people. We have DNA linked to electronic health records now. And the average follow-up is about 6 years. Average person has 100 billing codes, 600 labs, 600 drugs, 130 notes, and 10 radiology tests. And these kinds of prospective collection where the DNA is captured whenever they sort of agree to be part of the program, allows us to catch the really rare serious adverse events as well that happen that you couldn't necessarily collect as easily in a retrospective fashion. On the right here is a slide showing a way of looking at the validity of EHRs for replicating known associations. This is done using a kind of a fast way of developing phenotypes that we call phenome-wide association studies. And each row here represents a distinct phenotype that we are attempting to replicate. Each dot represents a distinct single nucleotide polymorphism genetic variant associated with that phenotype. And what you can see is everything to the right of that light blue line is a replication. So overall, we replicated 66% of those things using this kind of fast way of phenotyping from a record that we would have adequately powered for, and overall replicated 210 known associations and discovered about 70 new associations in this experiment alone. And the that study was part of studies done in the Electronic Medical Records and Genomics Network, which is a site of a network of 10 sites that have DNA-linked biobanks. And this shows you where they are. And we do a lot of studies within the electronic health record. And when we want to do a study in the EHR, I apologize for some of the formatting Mac PC issues on this slide. But what we see is we start with a phenotype of interest, and we develop an algorithm, which is an iterative process where you pull in different things like billing codes and medications and lab tests, and then you review it and revise it until you get to some threshold of positive predictive value. Then you deploy it across a site and maybe other sites such as an eMERGE, and do your genetic association test with your DNA. And overall, what we found is these algorithms almost always include billing codes, but usually also need to get at some evidence in the medical record, such as the notes, medications or important markers, and then laboratory and test results. And combining these with certain algorithms gets you to a high positive predictive value. I'll just give you one example of a common disease we did study analysis in eMERGE. And so this was after we first started, and we collected each site maybe a couple thousand cases that we had genotyped. And then we decided to look at another study. In this case, that was autoimmune hypothyroidism. And so we identified those cases and controls. We deployed an algorithm across the 5 different sites, and then put the cases that we identified at each site together, and we identified a signal. It's a thyroid transcription factor. Now, this is what that algorithm looked like. You can see that we have billing codes, and we have lab tests, and we use medications. We have some elements of time here that they have to have 2 of these things at least 3 months apart. And we looked at potential confounders like contrast exposure or medications that could alter your thyroid function. And here are some examples of things that are maybe a little bit rarer. One looks at an adverse event with ACE inhibitors, one of the more commonly used antihypertensive class of medications, and looking at the common adverse effect of cough. And we did natural language processing to find people that were mentioned in the notes as having cough while on ACE inhibitors. And we identified a potassium channel that was associated with that. And then a pretty rare disease, heparin-induced thrombocytopenia. Within our database of BioView at the time, which had about 100 and maybe 30,000 people in it, we found 69 cases of heparin-induced thrombocytopenia. And so to find these cases, we used an automated algorithm, but the automated algorithm wasn't good enough. And because it's such a rare event, you don't want to throw away any possible cases. So we then went in and manually reviewed anyone that looked like they might be a case of heparin-induced thrombocytopenia. And importantly, HIT has a pretty high mortality rate. If you're not familiar, it can be as high as a 50% mortality rate for true HIT. And so and then we identified a gene that was associated there as well. And we're doing other work on HIT now, focusing more in the HLA region. So that is a model that I think is, kind of points towards what we can do with other rare events where you really don't want to lose any cases. And so this summarizes some of the different phenotypes we've done in eMERGE. Overall, we've discovered everything in bold has a significant finding, and the ones with asterisks are new results. And I just want to highlight some of the rarer events in here. Multiple sclerosis progression from relapsing remitting to secondary progressive. Not a whole lot, not a great number of cases there. Warfarin-related bleeds, which we found a signal with, and drug-induced liver injury, which is also a pretty rare phenotype. That did not have a significant signal. Overall, this looks at all the algorithms that we have posted and collected at Vanderbilt so far across eMERGE and other networks. It's about 88 different phenotypes, and you can see the performance is generally pretty good, represented by the red diamond there. That's the median performance. But when you look at the rarer phenotypes, in this case drug-induced liver injury, the performance is lower, and that's, I would argue, exactly what you want, because you don't want to lose any cases. And when you look at SJS-TEN in BioView, this is work Elizabeth Phillips and Sarah Gannon and others have done. You can look at starting with the principle of using billing codes. I'm just showing ICD-9 here. ICD-10 is similar, but with different letters and numbers to identify them. Before 2008, there wasn't a specific code for Stevens-Johnson syndrome or TEN. And so you had a generic code, 1695.1, and then after that you had specific codes that came out to use, which had variable penetration and use, we have found. And then ICD-10 does have those specific codes. When you're thinking about really identifying and phenotyping in the EHR, in a lot of cases we actually have images now in the electronic health record systems, and that can help verify a given condition. However, that doesn't work if you have a de-identified biobank like we have. We actually don't have those images in the de-identified part because we can't certify that they are non-identifying. And then keywords are ways to search for things. And one of the things that Dr. Phillips is doing is collecting cases on an ongoing basis that have severe skin reactions. And so what we did is we actually built a real-time listener that monitors notes as they're written for keywords that are relevant to Stevens-Johnson syndrome, TEN, and can send, with IRB approval, alerts to certain providers to see if they are interested in being a part of the clinical trial. And that is identified, we launched that in around January, and has identified a handful of cases that weren't otherwise known. So they've done analyses and looking at phenotyping SJS-TEN in the EHR and putting these elements together against a number of specific medications and then validating them with chart review. And these are some of the results, and this is a poster that will be presented March 6th, and that you can see. In this case, I'm showing the data just for phenytoin, and the algorithm used at phenytoin use, and then ICD-9 10 code specific for either Stevens-Johnson TEN or for there's specific codes for rashes with drugs. And so putting those two together is the phenotype. And you can see the true positives of that collaborative algorithm here of those different components was 38%. And amongst that specific algorithm only missed 3 cases. Now they reviewed a much larger set and found all those people, and then done a genetic analysis on it as well. And then I wanted to think about a little bit about what some of the exposures are of medications that can cause SJS-TEN, to think about opportunities for intervening at the point of care for prescribing, asking the question of are these rare exposures, or if we had genotype-driven therapy for SJS-TEN, would we be making prescribing decisions and altering them? So we looked at 98,000 people who are medical home patients at Vanderbilt, and then looked at their exposure to just a handful of drugs. You see the list there: allopurinol, lamotrigine, phenytoin, carbamazepine, and abacavir. And these had at least 3 years of contact, and basically we found 12% of this population took at least one of these medications, and 6% took more than one, which argues that sort of 6% argues maybe for testing, when you do test, testing across a variety of HLA types, as opposed to just maybe the indication for one particular drug. And then, so after we find a population, you have the ability to, of course, look at things like comorbidity disease, what the outcomes are, longitudinally, evaluate for treatments and their efficacy, and then find the controls, of course, with drug exposure to do the sorts of genetic analyses which we're doing now. And finally, I just wanted to conclude with a sort of thinking about, where and efforts that are happening. Many of you are probably familiar with the Precision Medicine Initiative, which is looking to collect at least 1 million people from across the U.S. Really with, it's an sort of all-comers eventually. It's going to start with adults, but these people will agree to be as participants in the program, be recontactable, and share biospecimens and electronic health record data. So it will become another resource, just as with the various electronic health record biobanks, such as BioView and others that we've talked about, that will hopefully catalyze research in a number of domains. Again, it's not disease specific, but it will certainly, events that happen over time as people enroll will be recorded just as they are in our local biobanks. So with that, I'm done. We're doing questions at the end, right?
We'll take questions at the end. Thank you, Josh. So our third and final speaker is Cynthia Sung. She's at the Singapore Health Sciences Authority, and is going to talk about the regulatory perspective there.
Okay, thank you to Elizabeth Phillips for the invitation to speak. So what I want to do is just talk about 2 cases that the Health Sciences Authority has spent a fair amount of time debating about pharmacogenomic screening, and hopefully share with you some of the benefits and actual challenges of implementing pharmacogenomic screening. Next slide. Let's see. How do I advance? Okay. Right. In 2006, the Health Sciences Authority, which is the regulatory, like the FDA of Singapore, they introduced a module into the workflow process of the public sector hospitals, so that would facilitate reporting by the doctors directly into HSA. And after implementing that, the number of reports that HSA got increased by 10-fold. So now about 20 to 25,000 reports are received each year to HSA, and about half of those are related to the skin and appendages. So if you looked at this breakdown by system organ class, that's the biggest class that's reported in. And carbamazepine and allopurinol are like among the top 3 drugs that are reported. And so my talk is going to focus on these 2 drugs. So for the 5 years before we had any regulatory recommendations on genotyping, we had 91 cases of carbamazepine, which made up 1/5 of the total number of cases of SJS-TEN. Likewise for allopurinol, it's not quite as many, 66 cases, which is 15% of SJS-T and DRESS, because allopurinol is associated also with DRESS cases. Now from the, of course, the seminal work from Taiwan that have been identified an important allele in the HLA region, 1502 with carbamazepine. And when we look at the population prevalence, it's quite common among the major ethnic groups that are in Singapore, the Chinese, Malay, and Indian. And you can see it particularly in the Malay that it's actually 1 in 4 Malays carry this allele. So overall, there's ethnicity-weighted prevalence of 14.9%. That's, I just want to point out, that's different than the allele frequency. This is the frequency of both heterozygotes and homozygotes. So for 5801, this is very common in the Chinese population, less so in the Malay and Indians, for an overall ethnicity-weighted population prevalence of 18%. Now, because of the HLA region is so polymorphic, we felt it was very important to validate these studies locally. When we first looked at this data, we thought, okay, and I think the FDA as well, thought these markers were a pan-Asian marker, but our colleagues in Japan told us that they don't have 1502, so we realized that it probably wasn't a pan-Asian marker. And so HSA set up a case control study coordinated with 4 different clinical centers in Singapore. And over 5 years, we were able to, it's very tedious, able to collect 13 cases and 26 controls for carbamazepine, and we verified the 1502 association. Then more recently, we had enough cases and controls to do the allopurinol genotyping, and also were able to verify that relationship as well, with only 6 cases and 12 controls. So from the prospective studies from Taiwan, we knew the sensitivity and specificity of these assays. And because it's very rare, the negative predictive value is nearly 100%, but the positive predictive value is actually quite low, 6% for carbamazepine and only 2% for allopurinol. So in debating this, we had a lot of discussions for carbamazepine with people, with neurologists. And fortunately, there are good alternative drugs for epilepsy and neuropathic pain, which are the indicated indications for carbamazepine. And these drugs have comparable efficacy. The story is quite different for allopurinol. Allopurinol is the first-line drug, so it has the best efficacy. Probenecid and benzbromarone are second-line drugs. Our randomized controlled trials show that probenecid does not have quite the efficacy of allopurinol, and also both of these drugs, probenecid and benzbromarone, are not indicated, are contraindicated for people with renal impairment. And there's actually quite a sizable proportion of people with chronic gout that have renal impairment. So if they're positive for 5801, they can't be given this drug. A more recent entry into the market is febuxostat. It acts by the same mechanism of allopurinol. It looks like it has comparable efficacy, so we're encouraged by this. The problem is that it's 40 times more expensive than allopurinol. And in Singapore, almost no insurance company that I'm aware of has a prescription benefit. So the population is extremely sensitive to the cost of the drugs. So when we ran a cost-effectiveness analysis, we found that even at a PPV of only 6% for carbamazepine, that we did come in under the $50,000 per quality-adjusted life year threshold for genotyping. So from a health systems perspective, we could deploy genotyping and on average get a benefit without costing above this amount of money. Unfortunately for allopurinol, we did not reach that threshold. And I just want to, this is described in more detail in a poster, but we did look at, besides just genotyping, we looked at other interventions like a safety program and or combinations of a safety program and genotyping to try and figure out who would, how we could drive down the rate of allopurinol, SJS, TEN, or DRESS. And the analysis showed that there were actually 3 interventions that we looked at that were dominated, meaning they were more expensive and less effective and therefore would not be preferred. And one of those was genotyping to direct all patients who were positive to probenecid. And we also tested febuxostat, so that was dominated. The 2 that were what's called undominated, meaning that there's a possibility they would become cost-effective, but we need to manage costs on those programs. One of them was to have a more intensive safety program for all people who go on allopurinol, and that came in at $79,000 per quality-adjusted life year. And we calculated that we would need to get the safety program down to under $35 per patient in order for that to be cost-effective. But so that means that your safety program can't include an additional doctor's visit, right? Because it's going to cost more than $35. And then the other one that was undominated was to use genotyping, and if the patient is positive for HLA-B5801, to first put them on probenecid, And then if they don't respond, then to put them into an intensive safety program. That could become cost effective if we could get the cost of the test under $95. Okay, so for carbamazepine, in April 2013, HSA, in conjunction with the Ministry of Health, came out with a recommendation that going forward, genotyping prior to initiation of carbamazepine in new patients of Asian ancestry would be considered standard of care. And the reason we had to go in with Ministry of Health is because HSA only regulates drug products. We're not supposed to veer into clinical practice. So we needed Ministry of Health to come in and make that declaration. And that's, I think, what really made the doctors sit up and pay attention. In 2016, we came out with our recommendation with allopurinol, saying that it's not required as standard of care, but it could be considered in patients who have other pre-existing risk factors. An important one that was looking like it was important for us to consider is renal impairment. And so because we had the cost-effective analysis, we had the evidence for Ministry of Health to come in with a subsidy, and they provided 75% subsidy for the subsidized class of patients. Unfortunately, because we didn't have the supporting evidence, they did not provide the subsidy for the HLA-B 5801. So, as I said, the regulatory agency really can only regulate health products, and this is done primarily through the drug label. So working with the manufacturers, we got the drug label changed for carbamazepine, that genotyping is highly recommended, whereas for allopurinol, we just reported the association as for information. But we still felt that it was important for there to be facilities in the country for the genotyping. And so we reached out to the diagnostic laboratories and made sure there was at least one laboratory that could provide a validated assay. When we gathered the information on cost and turnaround time, and that was provided in the Dear Healthcare Professional Letter so that doctors would know where to go, who to contact, and what to tell their patients about the cost. So what was the outcome? So the outcome was that in the 3 years after implementation, there have been no cases of carbamazepine-induced SJS-TEN in genotype patients. So we think this is a huge success. Previously, we were getting about 18 cases per year. For allopurinol, it's just been 1 year since our letter, and unfortunately, the number of cases has not changed. So previously, there were about 13 cases per year. Last year, there were 12. So that's been a bit frustrating for us, and we're trying to think about other ways, other interventions. I'd just like to highlight this paper that recently came out about renal impairment. They gave us some really strong evidence about having both HLA 5801 plus renal impairment as a very significant factor. And so that makes us think about whether that might be a subgroup that we do some genotyping, and we're working on that. So just to conclude, that in addition to the high odds ratio of a genetic association and high prevalence in a population, we also need to consider the positive predictive value and the availability of alternative medications when we come out with a genotyping recommendation. And our real concern is if we mandate genotyping for allopurinol patients, we'll push a lot of patients into less effective therapy and maybe worse gout outcomes. So it's always this trade-off. The health technology assessments were key in driving decisions about the subsidy, and we're thinking about possible measures to mitigate allopurinol scar. We've published a physician guide. I've got copies here and by my poster to try and increase awareness of how to recognize these different types of dermatologic reactions. We have one hospital that's testing out like a telehealth safety program with some just messaging patients who are starting on allopurinol and, telling them, are you seeing any skin reactions, mucosal reactions? And we're thinking about some enrichment strategies for selecting subgroups of patients, perhaps that those might be worthy of genotyping. So this is our group at HSA working on this, a group of 7 of us. We also have academic and research collaborations, Duke NUS Medical School, Public Health School, Pharmacology Department, and the Singapore Immunology Network, which is doing our genotyping. And then I just want to also thank all of our clinical investigators who are helping us with our case control collection at 4 different centers. Thank you.
Great. Okay, so now we are going to transition to the questions session, I believe. And so if people have questions or comments, please come up to the microphone. I'll take advantage of moderator's role and maybe just ask 2 quick questions, if I could.
I had a few questions. I think what
Oh, okay. No. So one.
For the last intervention, my question, because it's more fresh in my memory.
Okay.
Last intervention for allopurinol, I think the position is ambiguous, because you, in the notice of the drug, you say genetic associations are reported and genetic tested is not recommended. So
it's not
what
that
does
it's
that mean?
not recommended, is that we're just, right now we just provide the information and it's up to the doctors to decide, where it's not at a system level.
Well, right, but what does that mean practically? If a patient has denied the test and get SGS UTN to allopurinol, he can turn against the prescribing physician, because in the notice it is said that SGS and TN may happen. You understand my point?
So you're saying that it actually gives...
You should take as a regulatory agency, you should take the responsibility of saying no test is needed, nothing more, or strong association has been documented and this test is needed. If you say, my lecture of your notice was genetic associations are reported and no test is recommended, it's up to you. You make the test or you don't make the test. That means that you transfer the responsibility of the decision to whom? To the prescribing physician and maybe to the patient also. I think it's not a good attitude. You should say, you should take your responsibility. If the association is not worthwhile taking in consideration, don't speak of it and do not recommend any testing. You understand my point?
We debated this with the rheumatologist and the dermatologist. This is what, with the regulators, the dermatologist and the rheumatologist say they still want the final to say. And so we've said it's not recommended as standard of care, but the doctors are the ones who said they still wanted the right to make the decision.
Maybe in your country there is no lawyers, because I mean that in the US, a notice like that, and a patient being not tested and getting TN from allopurinol would suit the physician and maybe also the federal agency who is making statements as ambiguous as that.
Well, but we also have to think about a bigger perspective is the gout population. So if we say you must test, and out of 100, 98 of them get less good gout treatment, we also have to think about that as an outcome. It's not easy. And that's why we're trying to think about how we can maybe through non-genetic risk factors identify the people who really should be tested, so we don't perturb the system so much away from proper gout treatment, but we find those cases who are most likely to benefit. It's a work in progress.
All right, thank you.
I do have a question about electronic, sorry.
No, you have one follow-up question?
About electronic records to look at epidemiology of SGS-TN. I had a very short and very interesting experience in my country, France. It's certainly different in other countries. As soon as the payment of the hospitals were related to the ICD-9 or ICD-10 classification, the ratio of TEN to SGS has grown tremendously. Meaning, I think that if you don't have a sample for controlling the reality of the coding, you are exposed to overestimate a lot because it's a natural trend of physicians. They don't know it's borderline, it's a 15% detachment. Who cares? TN.
Thank you.
I think that you cannot use that for example, for pharmacogenetic studies because the diagnosis is no more credible than in many other sources.
Josh, do you want to, I think, Josh, you're chomping at the bit to respond to that.
Am I supposed to? I guess it's working. All right. So I completely agree. So you have, interestingly, there's not necessarily a direct payment associated with SJS-TEN. In fact, in general, you have both false positive and false negatives, and I think the conclusion so far is mostly false negatives, right? People that don't have the code who have SJS-TEN. So it's a little bit of the opposite problem currently. But I would just emphasize that the findings of the results so far with this study has been you really do have to validate every case you find. This just helps you narrow down your set so you don't have to, you start with a set of 230,000 patients and you can find the real cases that when you look at the notes from consultants and verify that really is what you think it is.
Omar Iqbal from Loyola University Medical Center. While the researchers are investigating and finding new genomic markers, and while physicians are treating the adverse reactions, more and more drugs are being introduced. There is a huge number of drugs in the pipeline. Perhaps the FDA should be involved in analysis of these drugs, perhaps by chem-informatic aided pharmacovigilance, so that the drugs before they are introduced into the market should be tested whether or not they are having any toxicophores involved which could trigger an SGS reaction, rather than investigating at length on all different aspects. And while physicians are worried treating these patients, perhaps there has to be a control, because there are more drugs to be introduced into the market. Does it mean to say that the disease as such, it is called as a disease continuum? So does it mean to say that there is a research continuum, that this has to continue forever? There has to be a control over these drugs.
I think that a drug can be approved with, say, a safety database of about 3,000 patients. And so because this is rare, I think you may not even pick up one case, and that's why you have to really do the post-market surveillance. I think we won't really find very many in the pre-market space. But maybe, Jessica, you could comment on that.
I'm not aware, unfortunately, of any non-clinical biomarkers that we can use to identify pre-marketing. The only thing I could point you to, I know Keith Burkhart from FDA, as well as another person from the Office of Clinical Pharmacology, and David Jackson from Molecular Health used a program. It used to be called MAZE, but now it's called MH Effect, where they looked at drugs, the top drugs associated with SJS in FAERS database, and they looked for similar structures, targets, metabolic pathways. And in my slides, in the backup slides, there is the reference to that. But unfortunately, I don't think we have biomarker information or anything at this time.
I have a question, Dr. Weintraub, before we move on. I was just wondering if FDA has ever considered the idea of sending out buccal swab or saliva kits to newly reported cases of SJS and TEN, just to slowly but sort of over time accrue a biorepository that could be used to stimulate genetic research in the future as you accrue additional cases. I know that the vaccine adverse event reporting system attempted this in two situations, and they were really unsuccessful, largely due to the long time lag where they started the study many, many years after the cases were reported. But if you had some sort of system where you could tell that, maybe I've written a commentary on this, if you had a system where maybe you sent out the samples right away, you might get better compliance. Have you ever thought about that?
You know, I have not heard of anything along those lines, but I do know that we are using post-marketing requirements and post-marketing commitments in cases where we have recommended that genome-wide association studies have been recommended in order to further identify. So we have, I mean, I guess in effect, it's kind of pushing it off to the drug manufacturers, but we have used those post-marketing requirements and post-marketing commitments to further study.
Earlier, there was a discussion about whether there could be like an SOP for managing these patients, and maybe we should add to that SOP, collect a DNA sample. And
thank you. Just an anecdote, first of all, with regard to electronic health records. So I was asked to look at 150 cases of SJS-TEN from a well-known US provider, which will remain unnamed. And they were all coded as SJS or TEN, and none of the 150 actually had SJS or TEN. So there is difficulties with the way things are coded. And obviously that will happen in every country, but there is an issue. Can I just ask a specific question of Jessica, please? With regard to trying to identify cases and look at the burden of SJS-TEN in the US population, have you at the FDA used social media to be able to mine for cases of SJS-TEN and what drugs are implicated?
I'm not aware that we've used it for SJS-TEN, but we are, and I know the drug manufacturers are also looking at social media and text mining both in social media and in the published medical literature. So that is definitely something that we're looking at to the future, but it also comes back to the quality of the cases, and so it's sometimes difficult data to use, but I think, you know, we're not ignoring it.
Just one quick question, really, I guess, for the audience. Are there any countries that have mandatory reporting of SJS cases? I mean, we heard about the situation in Singapore where it's
No.
sort of built in, at least to the public hospitals, to facilitate it, but
right,
I
but
was
it's
just
still
curious.
not mandatory. It's still not mandatory.
Just maybe to answer your question, in Germany we do not have a mandatory reporting, and our equivalent of the FDA, the BfArM, Federal Institute for Drugs and Medical Devices, does not get reports of the cases. We exchange some data, and they get one case in 10 years. But fortunately, because we provide help for physicians treating these patients, information, discussion of the case, whatever they have, and we go and see the patients in the site, we have a very high capture rate of in-hospital cases, and all of these are ending up in hospital. So our recent analysis said that it's more than 90% of the patients that we capture that are hospitalized in Germany with SJS and TN.
I should say, though, that I think pharma is required to report it if they become aware of it. Is that correct? So I know maybe for the people, the patients who are here, that might be a route to getting that become more visible to the FDA, is telling the drug manufacturer this was a case, and then they are mandated to report that.
That's correct, and I appreciate Dr. Arden Jones putting in the plug to, you know, highlighting, report it to your regulatory authority or the drug manufacturer, especially for an event that is unexpected or unlabeled. We really, you know, without those reports, you know, we can't get the word out. So, so that's definitely one important thing. Let's see, I think there are 3 countries worldwide, I believe, that have mandatory reporting requirements. I don't remember which countries they are. I believe Sweden is one of them. Spain? Okay. I don't know that that's the answer. I think if you talk to people in that country, they'll tell you that there's still vast underreporting. I think Japan has an interesting system, and if I'm misrepresenting, I apologize, but I believe that the drug manufacturers are required to contract with some prescribers and users of the product for a certain number of years post-marketing to kind of to provide reporting of the products. And I think that's also an interesting model. So.
Canada as well, institutional mandated reporting, but it's just they don't have legislative to get it's passed by Parliament.
I'm going to come back to Munir's point, which I think is a really good point, and probably something that maybe I mischaracterized some in my talk. I want to make sure that the message is, we're not looking, in that particular example of phenytoin, it is the combination of, specific codes with drug exposure, with temporality, and also looking at, codes tying the adverse event to the drug itself. And so all the phenotype algorithms that work reasonably well have a pretty arduous process of validation against the medical record where you go against formal criteria. And I bet Katie and Elizabeth have spent a year working on this. So which is a lot less time than it would take to review 230,000 records manually. But it is not I'm not trying to suggest these are trivial processes. You really do have to dig into the record and read them. And that requires actually having a very robust EHR. And some EHRs probably don't have enough data that you can really read and understand what's happened to the patient and have that kind of data. So. It certainly can. Yep.
We actually did that just like phenytoin was drug specific, but when we did that, just looking at all cases carrying DNA that had those codes, and it was the predicted value of less than 20%. And if you look pre-2008 on a 695.1 code, then only 6% of those was 695.1.
And you have lots of false negatives. I mean, it's bad on both ends.
We're into our tea time right now. If it's a quick question, let's go ahead and then...
Very quickly, Simon Malal from Vanderbilt. Josh and I know each other well. So the real-time listener has been a phenomenal success. It not only identified cases that would otherwise been missed, there's been a rash of cases now found that would have been missed, I'd suspect, previously. But it also has been earlier. I mean, Dr. Phillips has been seen in the burn unit at 5 a.m. on Sunday morning now, so it brings the time forward. Earlier we had a discussion about the need to inform ophthalmology, gynae, so forth earlier. So that potentially could be a very useful mechanism for getting earlier engagement. But apart from getting the time of initiating the right expert to the patient sooner, is there a way of looking back in these EMRs to say, with machine learning, whether there were earlier signals that might have alerted people to the possibility of SJS? Because that could also have clinical translational benefit.
It certainly could. That's a great point. And, we are doing some work that as you guys know, where we're looking at clusters of symptoms and findings, although we haven't gotten all the details of those kinds of things in that would, we are seeing its efficacy to predict in advance of some things. We haven't tried this for, SJS, TEN, or other syndromes like, DRESS that would be very relevant. But certainly is on the docket. I think it is promising to think about the fact that the clusters of the components to making a diagnosis, though, certainly could lead to helping make diagnoses and suggest them to people who may not be thinking of them yet.
The one challenge you have with SJS is just training the algorithm properly. It's, as you know, you're sort of taking a very rare disease and then squaring it when you're talking about machine learning. So it's just it's a bit of a challenge unless there are incredibly distinctive attributes that are otherwise unrecognized. So, but I will take the opportunity to say that you actually made, I think, the first pun of the day with the rash of new cases, which means that it's time to draw everything to a close before we get a rash of more puns. Thank you.